Wow, I'm wondering if it's not a good. That's watching. All right, let's get started. All right, welcome back. We've got 3 weeks left. Right, we've got, and I wanted to. We don't have any results for exam 3 yet. I think they'll be posted by the end of the week. I expect we'll get them back in the next day or 2, and then we'll put up the curve, and you'll see where you're at. So I just wanted to throw out one thing to highlight. Today is like an undergraduate research symposium, basically lunchtime, 11 to 2 p.m. I know there's a lot of first and second years in this class, and if you're interested in doing research, this would be a great place to sort of see what's out there, see what other undergraduates are doing, find out what looks interesting, build a personal connection, build your research network, and then go out and work with, maybe, reach out to some of those PIs that are the senior authors on those posters and see if there's a position available. Because really, like, one of the USC strengths is their research programs for undergraduates specifically. A lot of great labs have students doing meaningful work there, getting published, and really jumpstarting their career. Whether you're pre-med or whether, even, I'd say even more importantly is if you're going into, if you want to go into like a PhD-based research program, best time to start is in your first and second year. Then you have like 2 or 3 years in which you can actually learn, how to do the most advanced techniques in the lab and really develop your skill set and put in enough effort to actually get results that will be published in that time frame that will be relevant to graduate school admissions. So that's at Roland Tudor's Campus Center, right? By where all the food is. in a ballroom, I think underneath in the basement, I think. Anyway, I'll be there for some period of time today, looking at my student stuff. All right. Anyway, so we've got 3 weeks left. We've got the reproductive system, and then we have the nervous and motor systems coming up. So next 2 lectures are reproductive system, and with a focus here on human stuff. But I think there is enough other systems out there, asexual reproduction, where it pays to know a little bit about how other animals do it. Okay. And here we have sort of a representation of various stages of development when you're in the womb, right? So this is human development, and we have in this sort of stage here, maybe 5 to 6 weeks post-gestation. The embryo's got this sort of curved body shape. You can already see like the developing sort of elements of it. We've got a neural tube, which is this sort of position down the back, which forms the brain and the spinal cord. We've got somites that are going to develop into vertebrate and skeletal muscle. At this time, our heart is starting to beat and pump blood. And then you've got the basic structures. The basic body plan is in place. Okay, we have maybe a tail that's going to get erased here. And then as we progress through development, okay, so this is sort of the embryonic stage. Then we're going to move on to the early fetal period. This is about 12 weeks in. This is a really important time in development where a lot of sort of genetic screenings take place during this time, or the nuchal test where people are looking for Down syndrome. And in this case, we have the fetus totally enclosed, okay, in the amniotic sac, which is this bag of fluid that surrounds the developing fetus. And we have already completed our organogenesis. So that is the formation of all the major organ systems within the body. That at least they're not fully developed, obviously, but they have formed. Okay, so you've had some sort of differentiation into that body plan. Onto that, we have external genitalia that are starting to differentiate. Your face is starting to look more like a human rather than a tadpole. And the heartbeat you can actually see via Doppler ultrasound. So that's a really important feature, right, to see whether or not the fetus is going to be viable. So this is why about 12 weeks in is a really important screening. And after that point, a lot of miscarriages generally happen before 12 weeks. And oftentimes people that are pregnant will wait until that time. They know they're pregnant, but they don't want to say anything until about 12 weeks in, because then the odds that you'll carry that baby to term are much higher than early on in pregnancy. Also, we have at this point the placenta, which is formed here. This is kind of a cool thing. So this is an organ that is associated with the uterus, okay, and it is what's going to be providing all the nutrients, all the oxygen through the umbilical cord here into the baby, right? So the baby's lungs are developing, right? But they can breathe in and out this fluid, right, to just exercise them, but they're not actually doing any significant gas exchange via their lungs. All of their sort of nutrients and gas exchanges coming in through this umbilical cord. And then finally, we've got the late fetal period here, about 8 months into development. And we've fully formed a fetus curled up inside the uterus, limbs are tucked in. What do we see? Our lungs at this point are mature. They've already been producing that surfactant that's going to allow breathing to take place. We've got fat deposits that are accumulating under the skin. The fat is going to provide thermal regulation. It's going to provide a substrate for you to sort of burn, energy, a substrate for energy after birth before you really are able to get sufficient milk to fully feed yourself, right? When a baby's first born, generally in the first 2 weeks, they lose weight. As it takes time for milk production to sort of ramp up. They can't, and their stomachs are too small to really bring in enough milk and colostrum to drive their development. And then finally, what else do we have? We've got coordinated movements. So a lot of the nervous systems and the brain organization that takes place is not fully specified by your genes, but has to have some sort of activity-dependent sort of competition between different sort of ensembles of neurons and how they interact with their muscles. So your body starts to move around, to kick, right? And then that aspect, it's one, exercising the muscles, and it's providing sufficient neural activity to sort of start mapping your brain in a way so that the appropriate neurons are connected to the appropriate parts of the body in an efficient and coordinated way. And then also at this time, this is where the baby's going to turn. Generally, they're supposed to turn. Doesn't always happen, but they try to turn and face head down, and that is going to start to provide pressure on the cervix, which will then start to develop the process of labor. Okay, so at this point, they're viable for birth, very high survival chances if they either are delivered normally or if they're delivered via cesarean section. Okay, so that's sort of like, some process, general overview of development here, but how do we start? What does the conception, moment of conception look like, right? And how do we get there? And what are the genetic strategies that are used by different creatures to reproduce their species? Okay, so that's really what we'll be talking about mostly today. So the key concepts that we're going to talk about are sexual versus asexual reproduction. Okay, there's different benefits and different mechanisms for both of them. The shapes and functions of all the reproductive organs, the most embarrassing part of the lecture. Then we have we've got the gametogenesis. So how are we going to form gametes? Those are like those are basically the germ cells, the cells that are going to come together to form the embryo, or the fertilized egg, the embryo in development. So how do we actually make those germ cells, both sperm and egg production? And they go in parallel processes, okay? Because actually, all of our, gonads and things like that are derived from, originally from the same sort of primordial organs, but there is some specialization, both sperm and egg, right? And the different strategies that sperm and egg have. Many sperm are produced, very few eggs are viable in any given month. And then how do our cycles of hormones going to control gonadal function, as well as the uterine menstrual cycles? Probably won't get that. So probably a lot of this might be in the second lecture. So let's talk about reproduction. We've got two major forms: sexual reproduction, asexual reproduction. What's the difference? Sexual reproduction is going to be the formation of offspring by the fusion of haploid gametes to form a diploid zygote. So we're going to go from in the humans 23 chromosomes to 46 chromosomes. We have to have a pairing. We have to have two copies of each chromosome, two copies of the genetic information that makes you. And that's why we always have two alleles of every gene within our body. And sometimes some genes are dominant, sometimes some genes are recessive, but we have two. Okay, and the process of asexual reproduction, that's going to be a formation of individuals where the genes are only going to come from a single parent. So sexual reproduction, two parents, each of which is giving you one of their two copies of their genes. Okay. Asexual reproduction, there's going to be no fusion of sperm and egg. There's going to be no searching for a mate. There's just going to be some very self-centered, very self-interested reproduction of the own body. So because of this different sort of design, mitosis and meiosis are involved in sexual reproduction, whereas asexual reproduction is going to be purely mitosis, purely just fission of cells. So asexual reproduction, lots of different ways this can work. A lot of invertebrates are going to use asexual reproduction, and many of them are going to, it's going to operate via fission. Okay, this is the splitting of an individual into two roughly equal halves. That's what fission is. This is actually when they just developed the sort of this understanding of biology and fission processes had just come out when people were trying to figure out how the atom was splitting apart, and when they were trying to sort of figure out how we might build an atomic bomb, what are nuclear physics, and they borrowed the term fission from biology to apply it to the physics in order to describe how a nucleus is going to split apart of an atom. So very similar. We have two fairly similar sized halves that are going to form. So you're going to basically make a double of each other. But that's not the only way you can do it. Another way is budding. Okay, so with budding, we've got this example here where you have a hydra, coral reefs do this. A bud is where you have a main sort of branch to the main parent body here, and then a new small part of that body starts developing into a unique individual. And well, I guess it's not a unique individual, a copy of the individual. And that bud then falls off and then can grow up into full size. So it's sort of an asymmetric splitting there, right? And finally, the next sort of stage is genules. This is where this is also his like some small parts of the main body coming off, but this is specialized in a way in that the genule is got sort of a special sort of shape. Usually it's in like a protective coating. It's almost like a seed that comes off, okay, that can then survive harsh environments. And when the environment is better, it can then turn into a full size, it can develop into a full size adult. We have fragmentation. Fragmentation happens, things like sea stars, starfish, they have like 5 arms. In this case, they can regenerate from broken fragments of themselves, but you have to have a large portion of that fragment. So in order for like a sea star to develop, it's got 5 arms, you have to have an entire arm plus part of the central core of that sea star. So if you break up, you can each sort of individual part of that can now redevelop into its own organism. Okay. And then finally, we've got parthenogenesis, which is a little bit more complicated form, which sometimes can happen with sexual reproduction as well. Parthenogenesis is where you are making eggs, but the eggs themselves can develop into, can sort of self-fertilize and develop into sort of its own individual. Okay. So what are some advantages of asexual reproduction? One, if you only have one individual around, okay, you don't have to find a partner. Okay, if I'm a lonely, the last turtle on Earth or the last turtle on the Galapagos Islands, he ends up dying because he can't find his partner because they rely on sexual reproduction. If there was a case that asexual reproduction was happening, that turtle could, create its copies of itself and continue to colonize Galapagos Islands. So this is going to work. So if you are down to a single example of a species, that you can still divide and sort of reconquer the area. 2, it works for fast colonization. If you are in an area where you're in an environment where you are highly adapted, where your genetics and your development, your body design is working really well for this particular environment, then you can very quickly colonize that environment and take over. Okay. The challenge with that is if that environment changes, you lack the genetic diversity for the species to continue to adapt to those changes, right? So if the environment changes, then you could have a complete catastrophe and wipe out that entire species. So let's now look at sexual reproduction. So sexual reproduction is where we are going to have fusion between an egg and a sperm. This is going to happen. Usually it's going to be a cyclical process in the sense that it's not always the case that an egg is available for fusion or for fertilization. Okay, it happens. It could happen with like photocycles. It could happen with seasonal cycles. It could happen with temperature. There's different ways that are going to regulate that. Oftentimes it's going to be driven by cyclical change in hormones as well. So human females have a cycle around 1 month, around 4 weeks, varies, that is going to allow a narrow window of time when fertilization can take place. You have all sorts of animals, other mammals, oftentimes will go into a period of like heat where they're only sexually receptive during this particular time. And that's also the only time that they can actually have eggs available for fertilization. Now, there are some mixed strategies in reproduction in some animals. Some animals use both asexual and sexual reproduction. The aphids have 2 kinds of eggs, okay, fertilized eggs. Well, if the eggs get fertilized, then they're going to follow a standard path of sexual reproduction. If the eggs remain unfertilized, then those can still develop into an identical copy of the female, okay, via parthenogenesis. So this is sort of like the best of both worlds, right? If you're aphid is like in an area where it's like really well adapted to the environment, but it doesn't have, it hasn't been fertilized, the eggs can still propagate the species there. But if there's are sufficient males around, you can fertilize and still get some shuffling of your DNA, which is great. It's adaptive. There's other examples where you have these sort of specialized, I guess, social structures, right? Within the honeybee systems, you have males, which are all drones. They cannot conceive, okay? And they're sterile and they are haploid. They only have single chromosomes. And then females come from fertilized eggs, and they are going to be able to, they're actually fertilized themselves, okay? And they're going to have diploid. Okay, so some terms of the art that we should know: gametes, okay, are eggs or sperms. These are the germ cells, okay? Zygotes, this is a fertilized egg. So right after fertilization takes place, where you have a fusion of sperm and egg, okay, it's going to now have a diploid, 2 copies of homologous chromosomes in the cell. And then if we have the germ cells, however, are going to be haploid that have only 1 copy of the homologous chromosomes in the cell. And so the process of, so inside our bodies, right, almost all of our cells are diploid, except for the sperm and the egg, right? Those are going to be haploid. We have to go through a process of meiosis to generate these sort of germ cells that are the eggs and the sperm, the gametes, that are missing half of the information, so that when we combine with the partner through fusion, we're going to have the appropriate set of chromosomes now to develop into a human. Okay, so sexual reproduction has got to enhance reproductive success over long periods of time, or survival, or it would rapidly disappear. It would rapidly get outcompeted by asexual reproduction, because asexual reproduction is going to grow exponentially faster than sexual reproduction. You know, if a single female splits here in asexual reproduction, you're going to make 2 copies through a sort of, say, a fission event. Each one of those can undergo fission. Each one of those can undergo fission. You're very rapidly increasing the total number of members of the population, right? In sexual reproduction, we're going to split, and that splitting is only going to produce half of the splitting. If you have 2 children, 2 offspring, right, you're going to have a female and a male offspring to then come together again and find a partner in order to continue to generate the line. So this is going to increase at a much lower rate. You need to have 2 offspring just to sort of maintain the overall propagation of the species. So there must be some advantage, right? And the main advantage is this genetic diversification of sexual reproduction, right? That is what's conveying the long-term adaptive advantages, because we are not in a static environment, right? The weather can change. There could be climate change, right? The corals are not doing so well in response to that, okay? There could be things where there's other species that are trying to kill you, or other pathogens that might want to kill you. COVID, some sort of crazy virus that wipes out most of the population, okay? If the genetic diversity of the population is very constrained, like an asexual reproduction, that can make you much more vulnerable to very specific attacks by, say, a pathogen, and can completely take out a species. So sexual reproduction maintains and actually enhances diversity within a population over time. And you can think of evolution actually as a way where we have our sort of species are continuously diversifying and shuffling our genetic material, continually making new mutations, and they're going to sort of propagate throughout the population over long periods of time. So we have these periods of great diversification, and then generally what will happen is there's going to be an evolutionary selective pressure that's applied. It could be a disease, it could be, you know, some sort of environmental change that will kill off a large portion of the population. And that is really what I think provides a lot of the evolutionary shifts in development across, you know, most eukaryotic species. Anyone that is produced by sexual reproduction. But that allows, because you have that diversification event before selective pressures occurred, this is how you're going to be able to maintain the overall species over long periods of time. And that is the key long-term adaptive advantage that makes sexual reproduction the dominant form of reproduction across eukaryotes. Most eukaryotes are going to use sexual reproduction, not asexual. Any questions on this stuff? Okay. So let's look at our reproduction systems. Okay, so there's two important functions they're going to do. They're going to produce the gametes, right? Those are these eggs and sperm that are going to fuse and develop into the individual. The reproductive system also are going to produce the sex hormones. So these are primarily produced in the gonads. They're secondarily produced in the adrenal glands. Okay, and the testes are going to produce sperm. So that's step one, the gamete, and it's going to produce testosterone. Ovaries are going to produce eggs, and then they're also going to produce female sex hormones, estrogen and progesterone. Okay, so as I've said, each male and female gamete is going to donate a single copy of each chromosome to the offspring. Okay, now in males are going to produce millions of sperm. Okay, and then try to deliver them to the female reproductive system. Whereas females are going to produce one egg on average each month, and then try to nourish and protect that egg to provide it as maximum amount of resources. So if it does get fertilized, it has the highest chance of turning into an embryo, a fetus, and eventually a baby. Okay, so each gamete is going to contain 1/2 the number of total chromosomes. We've got 23 total chromosomes here, and then each one of those is in a haploid organism. Each one is going to have one side of this. This is a karyotype. This is where you line up all the chromosomes based on how long they are. Okay. And so chromosomes, what are even chromosomes? They're just DNA material that's packed in and coiled up into secondary, tertiary, and quaternary structures. There's all sorts of things like histones that are grabbing onto those chromosomes and unwrapping particular areas of the chromosome in order to allow transcription to take place and genes to get turned on, essentially. But during the process of mitosis and meiosis and the cell division, these chromosomes condense into these shapes. Okay, oftentimes they're more strung out and like are all loopy, and you wouldn't even be able to see like them as a sort of a definite sort of finger like this. Okay, so each sperm has 1/2 of one of these. We've got 23 total. That includes the X and the Y chromosome. X is a female associated chromosome. Y is a male associated chromosome. And X is longer. That means there's actually more genes on the X chromosome than on the Y chromosome. So certain genes that are X-linked, if in males, you only have one allele of that gene, right? If it's missing on the Y chromosome. And this is why things like then certain things like eye color can be I'm not going to say. Okay, so when we have this fertilization event, we form a zygote. We have our full set of chromosomes, 23 pairs. Okay, 46 total. Now, so sexual reproduction. Why do we have it? The sexual reproduction is going to mix and shuffle genes from two parents to form this new individual, right? So we're taking one copy, okay, of each pair of those 23 chromosomes, but we also do a little shuffling of those pairs through homologous recombination during the process of meiosis. Okay, that is going to provide a greater genetic diversity to the mixing patterns that are brought down from each parent. Okay, we're going to make some new genetic combinations. Genes are going to interact in complicated ways that are going to produce different expressions, different phenotypes, right? Not everything is as simple as eye color. Like blue eye color is the result of a single genetic mutation, but hair color, body color, skin color, overall form of the body, basically any human trait, right, is generally going to be the combination of many different genes interacting together. Although many diseases can also be caused by a single mutation of a gene, either through a dominant gene that then asserts that protein that is screwed up, asserts a dominant influence over the other proteins that are produced by the body, or It's a combination of two recessive genes that are screwing up the same protein in the same place. Those things are going to be very much more rare diseases, probably. But when they're expressed, they can be very, very damaging and deadly. Okay, so our reproductive system is going to produce gametes, eggs, and sperm that are going to come together. How is that process going to take place? The gametes have to be haploid, right? They have to have half the number of chromosomes so that when the gametes combine, you'll have the full set. So this is the precursor cell, okay? This is a germ cell that's diploid, okay? It has pairs of homologous chromosomes, one from each parent. Okay. So we've got a red chromosome here. Let's make this red chromosome from mom. The blue is from dad. Look, they're the same length. Okay, so that means they're the same chromosome. Maybe this is chromosome number 1. It's really long. Maybe this is chromosome like 15 or something because it's sort of medium sized. Okay, so then there's going to be 46 total of these in here. 23 that are homologous to each other. 23 pairs of homologous chromosomes. Homologous means similar, but not identical. That is to say, they have the same genes on them, right? They're encoding the same set of genes, okay? But the actual alleles for those genes, the exact sequence of each gene is going to be different on each chromosome because one is from the mom and one is from the dad. Okay. Okay, so now what we're going to have to do is we've got to cut the number of these in half. Okay, now how are we going to cut the numbers in half? The first step that we're going to actually do is we're going to double the number, which just seems a little bit crazy, a little bit weird. What happens is we're going to make basically a copy. Each one of these chromosomes make a copy of itself and attaches itself to here. So this is still called a chromosome. Okay, this is called a chromosome. It's a piece of genetic material. This is also called a chromosome, but this chromosome has one chromatid, okay, and this one has two chromatids. Chromatid is a complete set of the genes for that one particular chromosome, and the sister chromatid is an exact copy, okay, one-to-one copy of the original one, and they stay attached to one another. Okay, so this is sort of the first stage of this development of either an egg or a sperm. Okay. Now what's going to happen? The nuclear envelope is going to dissolve. Okay. And then the homologous pairs here, that is to say the ones that have similar but not identical sequences are going to line up to one another. Okay. So chromosome 1 from the mom and dad are going to line up next to each other. Chromosome 15 from the mom and the dad are going to line up next to one another. Okay. Then you'll have a cell division. And in that cell division, half of these chromosomes are going to go to one cell and half are going to go to the other cell. Okay. And it's not a random selection. You don't get 2 of the 1s and like 2 of the 7s and 2 of the 13s and 0 of the 14s or something. If that happens, you're screwed up. You're not going to develop, right? Rather, this is done in a particular way so that you're going to get one chromosome from the mom and one chromosome, one each. For each of these 23 chromosomes, you're going to get one from the mom or one from the dad, but not 2 from the mom or not 2 from the dad, right? So you're going to get one each, and those are going to go over here. This is part of the shuffling, right? So now each cell has got 2 copies of one of each homologs. So it's haploid, but it has duplicated chromosomes here. These are identical to one another. Both sides of these chromatids, these are sister chromatids. These are sister chromatids. This one's from the mom, this one's from the dad. Okay, then to put the finishing touches, the second division in meiosis 2, to finalize the formation of the gametes. This is where these are then going to split sort of down the middle here, and you're going to get one copy of each sister chromatid. Okay, that's going to end up into each one of these germ cells. Okay. So what have we done in this process? We've created a tremendous amount of diversity, okay, in the set of traits and the set of genes that are going to be inherited from the parents, because you are basically taking a random selection of each of the 23 chromosomes. One is going to be from each one. You're going to basically roll a dice, okay? 50% chance I get my mom or my dad's for each one of the 23 chromosomes, okay? So that means on average, you're going to be 50% from one of the parents and 50% from the other, but actually you could inherit more from one parent than from the other parent. Okay. And that's going to create some of the diversity that you see across siblings, right? Now, you might think, well, but if I get a gene here on chromosome number 1, right, then I must also be getting the same gene I must be getting the same any gene along this chromatid, right? You might think would have to assort with any other genes that is located on this chromatid. Does that make sense? Because if the whole chromatid has to come with you, if you're only getting the one from your mom, right, then every gene on this chromatid has to come with it, right? So the diversity is actually being generated by which of the 23 you get, right? Or which of your male mothers or fathers you get out of each 23. That's where the main shuffling takes place. Or that would be the case if we didn't have a second aspect of shuffling, okay? The second aspect of shuffling is going to be homologous recombination, okay? So let's look at this homologous recombination. This is also known as genetic recombination. And this is how we can move genes and break the linkage between a gene that is on a single chromatid from a single parent. Okay, we can sort of shuffle that out of the population sometimes by this process of homologous recombination. So in this stage here, or our homologous pairs of chromosomes that have sister chromatids attached to them line up, what can happen along this interface between the homologous pairs, not between the two sister chromatids here that are identical. There's no sense in swapping them because they're absolutely identical, but you can get some crossing over events that are going to occur between these two chromatids, the ones that are homologous to each other but not identical. Okay, that's what it looks like. So we have a crossing over here, okay, and this is going to happen at particular regions of the genome that are identical to each other, and it's going to be facilitated by certain sort of enzymatic processes that want to facilitate that in order to enhance the genetic shuffling a little bit, okay? And the outcome of that will be the edge two chromatids are going to be precisely from the parent, the exact copy of the parent. The middle two will actually be a combination of both male and female parents, okay? So you have a 50, I guess you have a 25% chance of getting any one of these as your first chromosome, 25% for the second chromosome and whatnot. How often does that crossing over happen? It really depends on the length of the chromosome. They happen more often on longer chromosomes because there's just more material to cross over, but about 1 to 3 times per chromosome. Okay, so that gives you over many generations that actually allows maybe deleterious mutations to get sorted out of the population. Because if they were always going to be like this, then if you have one bad mutation here, right, on one of these chromatids, then it would always assort with all the same genes. So it would be very hard to ultimately remove it from the population. Okay, any questions about this? A little bit complicated. Okay, let's talk then about reproductive organs and gametogenesis in humans. Okay. So the sex organs in males and females are going to be derived from the same structures. Okay, there is a gene, SRY, that produces testes determining factor. And that gene gets activated around about 6 to 8 weeks after the formation of the fetus. And that is going to inhibit formation of female anatomical structural growth in males and enhance the development of male characteristics. Okay, so this is a key determining gene, and that is going to be associated with the Y chromosome. Okay. If you don't already know, right? Okay, we'll go into that in a second. Okay, so human male reproductive anatomy. Okay, so we've got external and internal organs, right? External organs are going to be the scrotum and the penis. Internal organs are the gonads. They're going to produce our sperm and our hormones. There's also going to be some accessory glands that will secrete the products that are needed for the movement of the sperm, for lubricating the ducts, and to like trigger things like uterine contraction. So the total ejaculate is going to be produced via a combination of sperm and these other secretions. Okay, so what's the overall goal here, right? We want to, in the male reproductive system, they want to produce sperm, lots and lots of sperm, millions of sperm every day, and then deliver it to the female reproductive system. So basically you have sperm producing devices down here in our testes, and then we've got a tube that's going to run past a bunch of different sort of glands that are then going to provide the combination of secretions that are going to allow support the sperm following ejaculation. Okay, so let's see here. So we've got, so the major elements here are going to be the gonads. Okay, those are the testes. Okay, and then we have the sort of sex accessories here. Okay, so we've got the epididymis, which is a sort of curvy, convoluted tubule, so to speak, that goes backwards and forwards, which is a place where the sperm are going to develop and be stored and progress through a series of development, and then that's going to then come down the vas deferens, which through a really, I guess, sort of random sort of path of evolution, loops up and over the urinary bladder, okay, and comes back down. It passes by 3 glands. We've got the prostate gland, we've got the bulbourethral gland, and we have the seminal vesicle here. Okay, each one of them is going to contribute different compounds to the ejaculate. Okay, so the urethra itself, then it's going to come out the urethra. It can be divided into the prostatic urethra, the bulbous urethra, and the penile urethra. Okay, and the vas deferens is what carries the sperm right here and is going to sort of wrap around. And then that merger here of this, let's see here, the vas deferens and the merger of that with the release from the seminal vesicles is going to contribute to is going to turn into the ejaculatory duct. Okay. Okay, so what are the accessory glands that you need to know? Prostate gland, okay. This is going to be where the prostaglandins were first isolated from, okay. And it's going to surround the upper portion of the urethra. It's going to produce citrate, which is a fuel for sperm. And also it's going to create alkaline secretions that are going to, raise the pH, buffer pH. That is going to activate sperm to drive their flagella, and it's also going to counteract the acidity of the female reproductive tract. Uterus and vaginal secretions are low pH in order to prevent things like yeast or other sort of pathogens from from growing and propagating within that area. So, so we want to sort of counteract that in the ejaculate. Okay, then we've got 2 conditions of our prostate gland that can happen. Okay, 1, over time, in age, that prostate gland tends to grow. Okay, it can constrict the flow of urine. Anybody that's stood next to a really old dude at a urinal knows that it might take them a long time to pee just to get going. That's because their prostate gland is probably sort of swollen. That also, because the prostate gland continues to potentially reproduce over the lifetime and grow like that, that can turn into cancer of the prostate. Okay. So that's usually detected through just feeling it, the rectal exam, or measurement of the acceleration of the pace of prostate-specific antigen. So there's an antigen that only the prostate gland creates, okay? The size of the prostate gland and the amount of division that's happening in the prostate gland is going to determine how much that prostate-specific antigen is circulating in the blood. Okay, now people have different sized prostate glands. There's no one number for PSA that is like you have cancer, but by longitudinally looking at it over time, the acceleration of PSA production is really the hallmark of prostate cancer. And that can grow uncontrollably. Then you need you can do surgical removal of the prostate. eventually testosterone ablation therapy, things like that in order to treat prostate cancer. Or immunotherapies are available now, where you train the dendritic cells within, you take out some infusion of your blood, train the dendritic cells to attack a prostate-specific antigen, reinfuse that into the body, and then you can mount an immune response against the prostate cells themselves. So any cancer that is derived from a prostate cell should express the prostate-specific antigen, and you can then train your immune system to kill the cancer wherever it has sort of traveled to. So that's sort of I think that was the first cancer immunotherapy that came to market by Dendreon about 15 years ago. Okay, so that's prostate gland. Now we've got our seminal vesicles. What are they going to do? They are going to release fructose. Okay, fructose, it's fuel, right? You can burn it. And the sperm have got to spend a lot of energy to get to where they're trying to go, to the egg. So that fructose within the ejaculate is going to be useful for them. Also, there's going to be amino acids that will thicken the semen to keep it from leaking, and also to assist the movement of the female reproductive tract. We're going to release prostaglandins. Okay, so the prostaglandins, remember, these fatty acids that are going to release and act as like paracrine signals that are going to trigger contraction of the uterus to draw in semen into the uterus itself. And then finally, we have bulbourethral glands. These are a pair of glands that are going to release basically a lube, okay, that's going to lube the tube and create this liquid before the ejaculate that's going to rinse off the tube, basically get rid of any like urine that's in the way, okay, and make sure that this sort of rather sticky stuff here, right, these amino acids and stuff, does not sort of clog, get stuck in the tube. Okay, so this is sort of a pre-lubrication. So those three things together are going to mix up into the ejaculate and be delivered into the female reproductive system. So what about the sperm? Okay, this is the actually most important part. This is our payload here. We want to get the DNA in there. The sperm is made in the testes, and they are surrounded by the scrotum, which is one of the two external elements here. Why do we have a scrotum? Okay, why isn't it just like, it seems like not a very safe place, okay, like hanging out there. The reason is because of how the DNA and the proteins that are made in sperm fold, and the optimal temperature for replication of sperm to prevent more DNA copy errors. It's better to have that happen at a lower temperature. Okay, so the scrotum removes the testes and keeps them further away from the core of the body in order to lower the temperature below 35 degrees centigrade. Okay. So if they are sort of, if they're produced above 35 degrees centigrade, they are going to prematurely activate. They might start swimming around. And temperature, an increase in temperature is actually a trigger that sperm are able to use in order to start their final process of activation and swimming. So we don't want that to happen prematurely, okay? The scrotum is sort of adjustable, right? Depending on external temperature, or like in a cold pool or something, that will try to pull, it'll contract, try to bring the sperm, or bring the testes closer into the body to maintain it in an optimal temperature range. If it's on a hot day or something, then they'll relax, try to keep the testes away from the body. Now these testes, okay, during development, they're made inside the body, and then at around 7 months of gestation, they will descend into the scrotal sac, okay? And you can see this here. Okay, so basically they're made here in the gonads, similar location to where the ovaries are going to stay, okay? But in the male reproductive system, it's this original, the ovaries and the testes come from the same primordial gonadal tissue, okay? They just differentiate based on that SRY. So gonad extends down the inguinal canal, okay? And eventually ends up into the scrotum, but it can get stuck at certain points, where you can have an inguinal hernia, where there's the inguinal canal comes out into the scrotum itself. Okay, and these are problems that can need to be relieved in order to surgically, in order to prevent sterility or a higher risk of testicular cancer. So this is called cryptorchidism if your testes does not fully descend into the scrotum. Okay, but surgically treatable. Okay, now let's look inside. Let's take a closer look at the testes in the seminiferous tubules where we make the sperm. Okay, so zooming in, okay, we've got this sort of convoluted tubule here of this of the epididymis, okay, that goes into the vas deferens. And before that, we have the testes, and there's all of these coiled up seminiferous tubules. Okay, this is where the spermatogenesis is going to occur inside those testes. And they're going to arise by this progressive differentiation of spermatogenic cells as they move into the lumen. So there's going to be sort of the primordial germ cells here on the outside edge of this seminiferous tubule. They're going to be surrounded by ligated Sertoli cells, which are going to be involved in sort of nurturing the development of those sperm. And as the process of meiosis and sort of differentiation progresses, they're just going to progress from the outer edge, where our sort of stem cells are located, and then progress into the lumen of this tubule. Once it's in the lumen of the tubule, then it's going to be made available. The sperm will be made available for ejaculate. So at different stages of this progression from the outer rim of the tubule to the inner lumen, we're going to have different counts of our chromosome count, different counts of, we're going to go from diploid to haploid, and different processes of division. In the outer edge, in these germinal cells, okay, they are going to continuously be undergoing mitosis, making copies of themselves. Okay, this is so that they are going to have to continue throughout life, throughout a male's life. They're going to always be making sort of maintaining these germinal cells. Okay, and in some cases they're just going to make the replicas of themselves. Other cases they might have a differentiation event. Okay, which will allow them to start the process of turning to primary spermatocytes. So in the primary spermatocyte, the chromosomes will double. Okay, you're going to make those sister chromatids. Okay, so and then they will, as we talked about, split up into these haploid cells under meiosis one in the secondary spermatocytes, but they still have 2 copies of each sister chromatid, so they need to split those. Okay, and now in the second division, the cells are going to become haploid. Okay. So that's going to be in our spermatotids, and then finally our mature sperm are haploid as well. Okay, so that's sort of, we can zoom in here and take another look at that, right? So our mitotic divisions are this self-renewing set of diploid chromosomes here in the spermatogonial stem cell. They're going to continue to renew themselves. Okay, sometimes that mitosis will result in this spermatogonium that then is going to mitose and split into primary spermatocytes. That is going to undergo meiosis one, split into secondary spermatocytes. They're haploid with pairs of sister chromatids, and then splitting the sister chromatids apart into 4 germ cells. Okay, so 4 gametes here. So one primary spermatocyte is going to make 4 gamete cells. Okay, those are then going to get packaged into the nucleus here of the sperm cell, and those mature sperm will be released into the lumen of the tubule. So this can take a long time. It takes about 2 to 3 months to make a sperm cell from that first division there. And it's going to really begin, okay, and then, okay, I said that. Okay. So spermatogenesis, we're going to change. Not only are we going to reduce the number of chromosomes to 1 member of each pair, we're going to change the shape. So we're going to be able to deliver the chromosomes. And right, I basically have said this. Okay, so these are just the terms of the art here, right? Spermatogonia, undifferentiated diploid cells, primary spermatocytes. We're going to have a diploid cell that's going to have basically it's going to be diploid and have sister chromatids. Then we're going to have secondary spermatocytes that are going to have sister chromatids, but only one pair of spermatotids are going to be haploid, and then we're going to split those into spermatozoa or sperm. Okay, let's take a look now at some sperm facts. 300 million sperm per day are going to be produced from puberty until death. We have this overall structure of the sperm that's going to be designed to reach and enter the egg. What do we have in that structure? Well, there's a tip here, okay, that has an acrosome. This acrosome is like this bag of or vesicle that has a bunch of digestive enzymes that is going to be involved in sort of digesting the coating around an egg in order to allow delivery of the payload, which is here in the nucleus, the DNA, into the egg. Then sort of below that, we have this mid piece here that's going to have a lot of mitochondria. Why do we need the mitochondria? We need to turn that fructose, we need to turn that citrate into ATP so we can turn this tail, the flagella. Okay, so there's a tail of the sperm. It's basically one long flagellum that's going to allow the sperm to swim to the egg. And this is the only cell in the human body that has a flagella. And this is what it looks like. You can actually, for a period of time, you can cut this off and the flagella will spin on itself as long as it still has some energy. And yeah, that's what it looks like. So what are the hormones that are then going to be involved in sperm production? It's going to be the same hormones that are involved in egg production. Okay, so luteinizing hormone and follicle stimulating hormone. These are both coming from the anterior pituitary gland. They're going to control both the rate of sperm production and the production of testosterone, right? Okay, so where does that come from? Okay, these are the gonadotropes of the anterior pituitary produce FSH and LH. And of course, there must be a gonadotropin releasing hormone. Okay, since we're in the anterior pituitary, there's always going to be a releasing hormone that controls the production of these hormones. So hypothalamus is going to make GnRH. GnRH is going to get released into that sort of those portal capillary network, and that's going to hit the anterior pituitary and regulate the production and release of FSH and LH. Okay, so they have distinct. They both are going to target the gonads, so the testes, but they're going to have distinct impacts on the testes. The first one, luteinizing hormone here, okay, is going to hit the Leydig cells. Okay, it is going to stimulate the release of testosterone from Leydig cells, and that testosterone will then, it's going to feed back and sort of inhibit GnRH release from the hypothalamus and LH from the anterior pituitary, but also locally within the testes themselves, it is going to promote the formation of sperm and spermatogenesis. Now, so FSH is a little bit different. FSH is going to stimulate it's going to stimulate these Sertoli cells, and the Sertoli cells will then themselves try to promote spermatogenesis. Okay. The Sertoli cells, the FSH is going to exert negative feedback via this production of inhibin within the testes. And the inhibin is what's going to feed back onto the anterior pituitary and try to inhibit FSH production. So if we don't have GnRH, okay, then FSH is not going to be released and Sertoli cells will not be activated, and you will have a negative impact on spermatogenesis. So thinking back to the endocrine system, anaerobic steroids can mimic that and can mimic the negative feedback here, and then that can cause negatively impact spermatogenesis and drive sterility. Okay, so what are these Sertoli and Leydig cells? Where are they? What really do they do? Okay, so we've got FSH, which is going to sort of promote activity of Sertoli cells. We have LH, that's regulating the Leydig cells. Okay, so Sertoli cells are also known as nurse cells. They're like going to nourish the sperm. They're going to provide certain nutrients that the sperm need, make sure that they're sort of growing appropriately. And then the Leydig cells are going to secrete testosterone. Okay, and that testosterone hormone is going to act as a paracrine signal to spermatogenesis. So they're in different places. So the Sertoli cells are actually within the tubule itself, adjacent to these spermatogonia and to the spermatocytes. Okay. Whereas the Leydig cells here are in the interstitial area on the outside of the tubule. Okay. And they're producing the testosterone, and that's going to travel locally. Remember, it's lipid soluble. Okay. It's a hydrophobic hormone. So it is actually pretty easy to transmit through this sort of tightly packed area of plasma membrane. Okay. So okay. So Sertoli cells are pretty important. They are going to phagocyte. Not only are they going to be releasing particular nutrients onto the spermatocytes to make the sperm, they're going to also phagocytose leftover cytosol that's around. They're also going to be involved in creating a barrier between the blood and the testes and maintaining the activity and propagation of this niche of stem cells that are along this outer edge. Okay. Any questions about sperm production? Okay. So we've made the sperm. How are we going to get the sperm where they need to go to create insemination and fertilization event? Okay, so it comes down to the other external reproductive organ, the penis. So sexual reproduction or sexual stimulation is going to lead to vasodilation of the blood vessels, this corpora cavernosa in the penis. Okay, so that is going to cause it to lengthen and stiffen here. And that is also going to then, the swelling of this sort of capillary network here is then going to also cut off veins that drain the penis. So there's sort of a positive feedback loop here. And positive feedback loops are common in all the sexual reproduction. Everything in the reproduction seems to be much more about positive feedback than negative feedback a lot of time. So we have the sort of dilation of these arteries via guanosine, cyclic guanosine monophosphate, or guanine, not, okay, sorry, cyclic GMP. Okay, that's what's going to cause the vasodilation of the arteries that are going to feed into the penis. Okay, and the sexual stimulation, it could be physical or mental, is going to stimulate nitric oxide production that triggers that cyclic GMP production. Okay, so we have some sort of sexual stimulus. It's going to cause release of nitric oxide, which then is going to bind onto this sort of pathway here that's going to allow a production of cyclic GMP and eventually vasodilation of the area. Okay, so how do we, there was a development of Viagra and then other Cialis, of variety of other sort of treatments for impotence, which is the inability to get an erection, which tends to happen in older males. So how does that actually work? So there is a enzyme called phosphodiesterase 5 here that inhibits the breakdown of cyclic GMP. It causes the breakdown of cyclic GMP. Okay, so this is an inhibitor of this enzyme. So this cyclic GMP then stays around longer and allows the maintenance of an erection for longer periods of time. Okay. Interestingly enough, like phosphodiesterase 5 is also found is the key sort of signaling molecule in the second messenger chain of your blue photoreceptors. Okay, so one of the side effects of Viagra is, at least at higher doses, is a blue tinge to the vision. In fact, in the original commercials for Viagra, all the commercials were in this blue hue. Nobody really knew why, but I think they were just sort of priming people for a potential side effect of that, because if you block phosphodiesterase 5 throughout the body, you're taking a drug, right? It can go anywhere within your body. Okay, it might work in the penis, it might work up in your head, it might work in your eyes, and that causes a prolongation of any signals from like blue photons hitting your photoreceptors. Okay, just like an example of how one drug can act on many different targets. Okay, so now we've got ejaculation, right? So the sperm has to get out of the penis. How is that going to take place? So they're going to be forcefully pushed past by a musculature. These are actual muscular tubules that can squeeze this and create a wave of sort of pressure that's going to propel the sperm up and out of the penis, okay? Past these different glands that are going to add those secretions that we've talked about before, seminal vesicles, prostate, bulbourethral glands, okay? And then eventually ejected into the vagina. Okay. So where does it go? Where are we going to try to merge the sperm with the egg? It's going to be in the female reproductive system, right? And this is the overall organization of the female reproductive system. The organs there are primarily internal organs. And we're going to have the pair of ovaries here, our primary gonads, right? And this is where eggs are produced and are stored prior to ovulation. Then we have the uterine or fallopian tubes here that are going to lead from these ovaries. They come down what's known as the oviduct into the uterus. And this is where the eggs are going to pass down through. Oftentimes the fertilization is going to occur within the oviduct itself, okay? And then we get down into the uterus, which is where the zygote will embed itself in the uterine wall and start to develop, right? And this is where the fetal gestation takes place, the development of the organs, the development of the body. And then finally, we have the vagina here, which is the sort of passageway for birth. Basically makes the birth canal. In between, we have the cervix, which is a tight closure of the uterus. And this cervix will dilate during, will actually get closed up by a mucus plug during pregnancy, and then will dilate during labor. The sort of level of dilation of the uterus tells you whether or not you are ready to actually deliver the baby. Okay, so let's look. Ovaries are similar in some ways and different in some ways to the testes. They are the site of oogenesis, which is for the production of eggs. Okay, so they're going to produce both the gametes, right? So our ova is ova are the eggs, or a single egg is called an ovum. And then also the hormones, they're going to be involved both in the production and regulation of the egg production and the development of the follicle, as well as the overall uterine or menstrual cycle. So oogenesis is different. It's only going to produce, instead of 4 viable germ cells, okay, it's going to, or gametes, it's going to only produce 1 per meiotic event. So at birth, every ovary contains about 400,000 oocytes. Okay, at birth, humans have already made all of the eggs that they will ever have. Okay, this happens during embryogenesis. Okay, and those are going to be sort of halted in meiosis and sort of they're going to be waiting there. Okay, and then at puberty, some of them, selected ones, just the ones that are just in the right perfect environment with the right perfect set of hormones that hit on it, are going to begin a process of development. Okay, most eggs are never going to be will never participate in ovulation. Okay, because you have 400,000 oocytes, you have about 1 egg that ovulates every month, and you've got 12 months in a year times, I don't know, 20 or 30. So not that many are going to make it, right? Maybe 1 in 1,000 make it. Yeah. So the steps of ovulation are as follows, right? So you have a bunch of these sort of primary follicles. These are eggs that are surrounded by a network of cells that are going to be responsive to fluctuations in hormones within the body. Okay, and those cells can grow and produce other hormones, these follicular cells, that are going to cause local regulation and promotion of the development, sort of positive feedback of the development of that primary follicle. These are actually going to compete with one another, each of these follicles, to see who can sort of grow the fastest or at the appropriate, reach the appropriate stage of follicular development at the time of ovulation. And one follicle will become the dominant follicle on any given particular cycle, any sort of month cycle. And this is why we're only going to have one released, generally only one release per cycle. Sometimes you'll get 2 different eggs will be released, but almost always it's going to be 1. Okay, so every primary follicle has, I'm going to have 1 egg, 1 oocyte. Okay, and it's going to be surrounded by granulosa cells and thecal cells. these are basically doing the same thing for the egg as Sertoli and Leydig cells do for sperm. Okay. And they are going to be regulated by the same hormones that regulate Sertoli cells and Leydig cells in the sperm. So LH, luteinizing hormone, and FSH, follicle stimulating hormone, from the anterior pituitary. What are they going to do? Okay, follicle stimulating hormone. Pretty easy to guess what it's going to do. It's going to stimulate the growth of follicles, okay, which are the primary follicles surrounding the egg. Okay, luteinizing hormone is a little bit more complicated, and it is going to actually influence the ovulation event itself. A surge in LH is going to be what triggers the rupturing of the follicle and the release of the oocyte into the oviduct. And it is also going to be involved in maintenance of this, corpus luteum, this yellow body, which is what's left over of the massive cells of the sort of fully developed follicle that ruptures. Those cells don't just die, they turn into a new sort of structure called the corpus luteum. You have one per cycle that is going to release hormones and potentially maintain pregnancy or eventually sort of degrade, depending on whether or not a fertilization event takes place. Yeah. So every month, only one follicle matures. The sort of overall process here of the passageway into becoming a mature follicle takes place in waves. Okay, it usually takes about 3 cycles for a primary follicle to fully develop into a mature follicle. So it's almost like a pipeline of follicles of different sizes that are sort of competing against one another. Only one gets to be dominant. The other ones will regress based on competition for hormonal resources within the ovary. Okay, so after release of the egg, it's going to be viable for about 24 hours. Okay, the sperms are going to be swimming around there for 3 to 5 days. Okay, so you have a window of time where you have to have an intersection of sperm and egg within the oviduct, typically, in order to allow a fertilization event to take place. If we get a fertilization event, now we're a zygote. We've combined 2 haploid germ cells into a diploid cell. And following that, this corpus luteum is going to turn into this empty follicle is going to turn into the corpus luteum. Okay, it'll regress if fertilization doesn't take place, and it releases estrogen and progesterone. So let's take a sort of zoomed in view here of these cells. Okay, so we have granulosa cells and thecal cells. Okay, so the granulosa cells are sort of like the Sertoli cells. They're close to the oocyte. They're going to be nourishing the oocyte. Okay, the thecal cells are going to be sort of further away. They're going to be involved in hormone production. Okay, so these granulosa cells, they are going to produce sex steroids. Okay, growth factors for the development of the oocyte. So FSH is going to stimulate the granulosa cells, and the granulosa cells, what they are going to do is actually they're going to convert androgens, testosterone that's produced by the thecal cells. Okay, they're going to actually convert that over into estradiol, which is the main estrogen female hormone, by an enzyme called aromatase. And they're basically taking testosterone produced by the thecal cells, turn it into estrogen, and that estrogen is then going to impact the menstrual cycle and the development of the oocyte. So here is where our nuclear material is. Okay, then we have this is where the DNA is found, right? We have this oocyte. This is the yolk itself. This is where you're going to have a lot of basically nutrients, right? So that you have enough material for the developing fetus to sort of grow through the first several cell divisions. And then surrounding this, we have this cumulus oophorus. Okay, these sort of cells are going to aid in the acrosomal reaction and allow the breaking down of this zona pellucida, which provides a barrier between the cell, between the sperm and the egg, right? It's important to only have one sperm fertilize the egg. You have to have only one copy you have to have, one set of haploid genes to combine with the ones that provided by the oocyte. If you had two sperm that both try to fertilize the same cell or both successfully fertilize the same cell, that embryo is not going to be viable. Won't even turn into an embryo. Okay, so now what else do we have? So we've got so thecal cells, as I said, they're going to synthesize androgens, testosterone. Okay, they are going to provide signal transduction between our granulosa cells and the oocytes during development. The LH is going to be, so luteinizing hormone stimulates the thecal cells to produce testosterone. Okay. And that testosterone will then sort of act as a paracrine signal on the granulosa cells that get converted to estrogen, which then acts on the surrounding tissue here, this cumulus oophorus, as well as the egg itself to further the follicular development. All right. So one other aspect of this zona pellucida, so this area surrounding here, this is where we're going to get sort of the species selectivity. So only, like, what makes a species, right? It's kind of a debate, okay? But one of the key factors that makes us, makes like one member of, like two individuals, the same member of a species, is whether they can interbreed with one another, okay? Like dogs have great genetic diversity, right? We've bred them in all these different ways, right? But they're all considered part of the same species. Because they can all breed together. The output might not be very good if like a Great Dane impregnates like a Shih Tzu or something, but that might not be good for the delivery of that dog. But they can create viable zygotes. Okay, what prevents that this crossbreeding from occurring is the composition of this zona pellucida. So the zona pellucida has a bunch of glycoproteins, so these sugar-based proteins that are going to bind spermatozoa, and that interaction is very specific to the species, to sort of binding molecules on the head of the sperm and there. So that binding is going to be a requirement for initiating this acrosome reaction that allows the digestion of the zona pellucida and the delivery of the genetic payload. Okay. So let's, I guess, this is, I think, a useful slide in the sense that it breaks down the cellular divisions of sperm on the right-hand side, right? And compares it to the cellular divisions in the developing oocyte. Okay, so remember, in our primordial germ cells, okay, these are creating our oogoniums, okay? And then these oogoniums are going to sort of divide via mitosis and turn into primary oocytes. All of the primary oocytes are present at birth. Okay, this whole process basically stops by the time birth takes place. We have our full set of 400,000 primary oocytes, and those are all paused, prophase of meiosis 1, and then they're going to split. Okay, in meiosis, we're going to split our chromosomes, our set of chromosomes, and unlike in sperm, where you have a symmetric division here, in this process of oogenesis, we're going to have an asymmetric division. Okay, one is going to sort of one side is going to turn into what's known as a polar body. Okay, and the other side is going to turn into the secondary oocyte, and that also is going to be paused. This metaphase in meiosis, okay, until it is found within the environment of a mature follicle that has all this sort of cellular material around it, okay, or all these cells around it that are there to nourish it, provide an appropriate feedback, appropriate hormonal control of that, or hormonal development. And at the sort of moment of ovulation, this is going to complete. Ovulation is going to come down here. Okay, it's going to travel down, and at the moment of sperm entry, it triggers the second stage of meiosis 2. We're going to have a second division of this area into a second polar body. So why do we do that? Okay, so basically, this is throwing away one half of the genetic material here and another half of what's left right here. So that we're going to come down to a single sister chromatid and a set of 23 chromosomes right there. Yeah, and we're throwing away the material, but we're throwing it away in an asymmetric way so that all of the cellular resources, all of the yolks, we're not undergoing a symmetric division because we want to sort of maximize the total amount of nutrients available to the yolk, all of the mitochondria that might be in this yolk, all of that are going to be sort of packaged into one asymmetric side to maximize the ability for that egg to develop successfully. Okay. The other things are thrown away and digested. Okay, and so following that ovulation of the secondary oocyte, this ruptured follicle will develop into corpus luteum and either degrade or stay around for a while, depending on whether or not fertilization takes place. Okay, so I think I'll stop there. We'll continue tomorrow, or to continue on Thursday to finish up sexual reproduction and reproductive systems Q&A at 11:45 today on Zoom. Hello. Yeah.
The slide where you show the common films, yeah, is the